On behalf of Faculty of Pharmacy, I warmly welcome our respected principal, sir, distinguished speaker, esteemed faculty members, and our enthusiastic M.Pharm and B.Pharm students who have gathered here today. Faculty of Pharmacy, Dr. MGR Educational Research Institute, under the benevolence of our Honorable President, sir, and Honorable Secretary, sir, is organizing a series of webinars, FDP, and professional talks. We thank Professor Dr. S. Geeta Lakshmi, ma'am, Vice Chancellor, for her constant support and guidance. Scientific research forms the backbone of pharmaceutical innovation, and good laboratory practices plays a vital role in ensuring the reliability, quality, and integrity of research outcomes. Today's session will provide valuable insights into how GLP contributes to producing credible and reproducible scientific results. We are truly delighted for such an enriching academic session that will help strengthen the research skills of our students and scholars. To formally begin today's program, I would like to invite Dr. P. Senthilkumar sir, Professor and Head Department of Pharmaceutical Chemistry, to introduce the program and share a few words about the importance of the today's session. So we welcome you, sir.
Good morning to all. On behalf of Faculty of Pharmacy, it's my privilege to deliver the introductory address. I am very glad to welcome you all to online webinar on the topic of Skill Development Program and Good Laboratory Practices, the backbone of reliable research, organized by Faculty of Pharmacy, Dr. MGR Educational and Research Institute. Faculty of Pharmacy, Dr. MGR Educational and Research Institute is successfully organizing a sequence of national and international conferences, webinars, workshops, hands-on practice, and faculty development program with eminent speakers from all over the globe, with a vision intensify the intellectual wealth to the students, faculty, research scholars, industrialists, and pharmacists. In continuation with the events conducted, Faculty of Pharmacy, Dr. MGR Educational and Research Institute is organizing this webinar, which is emphasize and learn more about skill development program on good laboratory practices, the backbone of reliable research. This program is organized with the objective of enhancing the practical knowledge and laboratory skills of students and researchers by introducing them into the fundamental principles and standards of GLP. GLP is a crucial role is ensuring the quality, reliability, and integrity of the laboratory data in the field of pharmaceutical sciences and research, maintaining the proper laboratory procedures, documentation, safety measures, and ethical consideration. This program is help participants to understand importance of maintaining standard procedures and following the regulatory guidelines while performing the laboratory experiments. Though this webinar, participants will gain valuable insight into the laboratory safety, proper handling of chemicals and instruments, accurate documentation, and quality control practices. Such skills are essential for building competent professionals who can contribute effectively to the research and industry. At this moment, I would like to extend my heartfelt gratitude to our management, Founder, Chancellor, Dr. A.C. Sanmugam Sir, Honorable President, Engineer A.C.S. Arun Kumar Sir, Honorable Secretary, Thru A. Ravikumar Sir. Under those guidelines, all our efforts are taking in this shape. I would like opportunity to thank our Vice Chancellor, Dr. S. Geeta Lakshmi Ma'am, for his constant support and guidance. I also welcome and extend my heartfelt welcome to the speaker of today's webinar, Dr. R. Karthik, Head Bioanalytical and Bioanalytical Department, Sidus Pharma Service Private Limited, Chennai, who accept our invite to share his vision, the research backbone of GLP for pharmacy students. My sincere thanks to our beloved Principal Sir for his guidance and encouragement in all our efforts. I welcome all the participants, students, and faculty members present in this program. So let's gain the knowledge in the extreme interesting topic, GLP, the backbone of reliable research. Thank you.
Thank you, sir, for that inspiring introduction and for highlighting the importance of today's program. Now, I kindly request Dr. S. Nandakumar sir, Professor and Head Department of Pharmaceutics, to kindly deliver the welcome address.
Thank you, Ms. Nilambari. Very good morning to all of you who have gathered here on this online platform. It gives me a great pleasure to welcome you all to this skill development program on good laboratory practice. As Dr. Senthil Kumar sir has already informed, I would like to, register here our thankfulness and gratitude to our management who have been always very supportive in conducting all these activities to our students. And my regards to our beloved principal, for motivating us in all these endeavors. And my special thanks and my special welcome to Dr. R. Karthik, Head of Bioanalytical Department, Citus Pharma, for having accepted our invitation and being here to deliver the topic on good laboratory practices. Probably a very important topic for all the departments. Not only to analysis, for all the other departments also. So this is a very, as the title itself says, it's a very important, the backbone of reliable research. So thank you for being here, sir, and I thank you in advance for for being you are going to enlighten our students. And to the students who have gathered here on this platform, I would like you all to take this platform very seriously, hear out from our resource person, and please clarify your doubts so that people who practice this are the best people to teach you. So that is the real motto why our institution is bringing up all the experts from industry to teach you all these topics. So I request you all to utilize this opportunity to the best. And with that note, all the best to all of them who have gathered here. My special acknowledgement and regards to the head and the faculty members of Department of Analysis for organizing this event. Thank you very much.
Thank you, sir, for your motivating and encouraging words. Now, I kindly request Dr. S. Nandakumar, sir, Vice Principal Admin, Professor and Head of Pharmaceutics, to felicitate our esteemed speaker as a token of gratitude.
Dear Dr. Karthik, kindly take our token of gratitude as the certificate of appreciation for your contribution. Thank you very much, sir.
Thank you, sir.
Thank you, sir. Thank you, sir. Every successful academic event becomes meaningful through the knowledge shared by distinguished experts. Today, we are fortunate to have an accomplished professional who has extensive experience in the field of pharmaceutical analysis and bioanalytical research. To introduce our respected speaker, I invite Mrs. P. Soundarya Madam, Assistant Professor, Department of Pharmacognosy, to introduce the expert of the day. We welcome you, ma'am.
A very good morning to all. It's my immense pleasure to introduce our distinguished speaker, Dr. Karthik Rajendran sir, today in this occasion. Dr. Karthik sir is a pharmaceutical research professional associated with Citus Pharma Services Private Limited, a contract research organization based in Chennai. Citus Pharma Services Private Limited is one of the leading contract research organizations located in Chennai in the southern part, delivering services with quality, ethics, and compliance to the standards set by the major regulatory agencies around the world. Sir is associated with Citus Pharma Services Private Limited as Head Bioanalytical and Analytical Department from November 2017 till date. Within the organization, Sir has been involved in development and validation of bioanalytical methods like UPLC, mass spectroscopy techniques, supporting bioequivalence and bioavailability studies for pharmaceutical products, ensuring compliance with regulatory guidelines such as US FDA and global standards, supervising laboratory operations and analytical testing teams, collaborating with research institutions and pharmaceutical companies. Sir has worked previously with Apotex Research Private Limited Bangalore for almost 11 years, wherein he has played various roles from bioanalytical scientist, senior bioanalytical scientist to team leader and group leader. Sir has also worked with GVK Biosciences and WOCAD Limited previously. Dr. Karthik sir has been awarded PhD in Pharmaceutical Sciences from JSS Academy of Higher Education and Research, Karnataka. Sir has undergone his undergraduation and post-graduation in pharmacy at Tamil Nadu Dr. MGR University, Chennai. Karthik sir has been involved in scientific publications and conference research, particularly in pharmaceutical bioanalysis. Example, research on UPLC mass spectroscopy analytical methods for quantifying drugs such as carbamazepine and its metabolites in human plasma used in bioequivalent studies. Such work helps improve accuracy of drug concentration measurement, efficiency of clinical pharmacokinetic studies, regulatory compliance in drug approval processes. His key achievements include leadership in bioanalytical research with a CRO environment, contribution to validated analytical methods for drug studies used in clinical research, has handled 300 plus molecules method development and validation, handled 7 which were submitted to the regulatory of US FDA, MHRA, EMA, TPD, TGA, GCC, NMPA China, and NPRA Malaysia. Has handled 30 plus molecules for NDSRI, nitrosamine impurity, and elemental analysis in drug substances, drug products, and excipients, which were submitted to US FDA. Has successfully faced US FDA, GLP, GMP audit, MHRA, EMA, and GCC audit with no observation, and visa audit with recommendations. Participation in numerous pharmaceutical science conferences and research publications, supporting clinical research products that help pharmaceutical companies obtain global regulatory approvals. Through his work, he contributes to the development of safe and effective pharmaceutical products and supports broader drug development life cycle. And finally, we are pleased to have you here, sir, for the skill development program on good laboratory practices, the backbone of reliable research. Thank you.
Thank you, ma'am, for that wonderful introduction. Now we come to the most anticipated part of today's program, the expert talk. It's my honor to welcome Dr. R. Karthik sir, Head of Bioanalytical and Analytical Department at Citus Pharma Services Private Limited, Chennai. Sir brings with him rich industry experience and profound expertise in analytical research and laboratory practices. We are confident that today's session will greatly benefit our students by providing valuable practical knowledge about good laboratory practices and their importance in ensuring reliable research outcomes. Sir, we are delighted to have you with us today. I kindly invite you to deliver your expert talk on good laboratory practices, the backbone of reliable research. Dear students, please join me in giving a warm welcome to our esteemed speaker. We welcome you, sir.
Thank you, ma'am. First is thank you for this introduction, and I thank Dr. MCR Educational Research Institute and Faculty of Pharmacy Principal, Vice Principal, and staffs, HOD of Chemistry and Analysis Department for organizing this skill development program. And it's my pleasure to deliver this topic related to GLP, and which is the backbone for the laboratory researchers and for getting the approval of the drugs. Not only for the drugs, GLP has been applicable for other applications also, rather than the pharmacy. So it's my pleasure to share my knowledges and what are the requirements it has been done in the principles and the practical examples. This is for graduates and postgraduates and research scholars as well. So next slide, please. Okay, so today's presentations, what we are going to learn is we will be going to understand about the GLP fundamentals and what the regulatory, how the GLP has been evolved through the regulations and practical implementations of the GLP and the scope where the GLPs have been utilized and what are the core principle and what are the guidelines as they have been revised from starting from 1997 to till 2024 or 25, whatever it is there, and recent updates and real world applications of this GLP. And once after this completion of all this, we will come to know the enhanced compliance competencies and how we are going to improve our study design based upon the GLP and how we are going to maintain the quality and the integrity of the data when we are going to develop the method or whatever we are going to do the research. So in that the quality and the integrity, how we are going to implement it, we will come to know and will be gain the knowledge about that one. So next slide, please. So first is the introduction. So what is a GLP? It's a GLP nothing but the good laboratory practices. So the definition itself, it will clearly explains what the expectation is from the regulatory and what the GLP is actually it is going to tell us. So the definition it says it's a managerial quality control system. It's covering the organizational process, conditions for planning, performing, monitoring, recording, reporting, and archiving of non-clinical safety studies. So GLP is applicable only for the non-clinical safety studies. So wherever the part, either it can be an agricultural or a chemical, or it can be a pesticide, so or it can be a pharmacy. So wherever we are doing any non-clinical safety studies, is and that particular product, if we are going to get an approval from other regulators like US FDA, Europe, they have to do perform the testings in the GLP approved lab. So the GLP approved lab, it has to be get approved, and then that particular approved lab, whatever we do it, and that is going to be get accepted by the regulatory agencies, including in India also. And here the 3, 4, 6 things is it has to be noted down. So whatever we are going to do, the activity or the study, it has to be planned properly, and the what we have planned, it has to be performed as per the plan, and it has to be monitored, recording, reporting. So this is the main definition, and the purpose of this GLP is it ensures the data, whatever the data which has been generated from the GLP lab, it ensures that the data is quality and the integrity it is there, and the reproducibility will be getting it from the results, and it gives the regulatory an impression that the data generated from that particular GLP lab is confidential, and it gives a correct data like that. So it will make the regulatory to decide it and give an approval for that one. So for that purpose, the GLP is important, and the regulatories which follows all GLP is OECD group of countries as FDA, EMA, WHO, and national authorities. So they frame this GLP and they give an approval for the products as whatever they do it, and they will ensure that they did it in the GLP lab, and they will ensure as per the principles all it has been conducted or not. And there is a distinctions between the three terminologies, that is GLP, GCP, and GLP. So GLP is governs only for the clinical laboratory studies, where GCP is for clinical trials, good clinical practices. It is there. And for manufacturing, if you go, it is a good manufacturing process. So it's all three are different principles and procedures. It is there, but each are been interlinked to each other. So next slide. So first we will be seeing the historical context and why the GLP it came. So what is the necessary for the GLP? So in early 1970s, it started the GLP, and there what happened there, when US FDA, when they do the inspections and to the data which they have been submitted, so they came to know that there is an inconsistency in the methodologies, or the documents which they have been submitted, there were inadequate documentations, and when they go for inspection, so they identify there is a data manipulations. So because of all these things, the need of a GLP it came. In 1970s, almost 40 US labs, they have been toxicological labs especially. So they went for an auditing of the facilities as the data which have been submitted. And they identified that out of this, the industrial biotest laboratory, so that is a very big company in US, and they do almost 80% of the studies whichever they conducted for the toxicological animal studies. They do it, and all 40 studies, 40 labs, USFDA, they went for thorough investigations and auditing, and they identified that a lot of poor practices they have been doing, like the equipments are not being properly calibrated, right, and they do it for analysis. So if the proper calibration is not being done, the results which they have been getting it from that particular instruments or the equipments, it may not be reliable. So like that it is there, and the persons has proper training. So before they execute any activities, they should be get trained to that particular activity or for the instruments. that is not been done. And proper qualified persons, it has to be taken, and there is no proper qualified persons are not there. And some cases they don't have a standard operating SOPs. That is standard operating procedures they don't have, and still they do it. And they don't maintain the test reference systems as that animals is how to handle it. And there is no proper segregations of the test and the references items. So like this, a lot of poor practices they have been identified, and on this, because of this, what happened in 1978, they identified all these doing as 80% of the studies which they have been conducted in IBT. So what they have been done, they have been tested lotions, deodorants in the mice, and what they did, those tests what they have been conducted, those mice has been died, but they have been not shown those things and without showing that they have released those products for the human users, and because of their manipulation that products are being used by the humans. So they identified this and they have imprisoned the owner of that particular company, and they have closed that particular company. And then USFDA, they have made it in 21 CFR Part 58 in 1978, the principles of GLP. So after that, in OECD, they adapted these principles in 1981, and they started using internationally these particular principles. Next slide. So here how it has been got evolved. So in 1978, USFDA, they brought the principles, and first they started using it, and based upon their principles, OECD, that is the Organization for Economic Cooperation and Development, they adapted these principles and they recommended their member of countries to follow this one. And among to that, the member of countries, apart from those who have been registered, and few more, they have been few countries they have agreement, that is mutual acceptance data. Those countries also they started following it. For almost 2 decades, they followed the OECD principles, and that 2 decades, what are the things as they have been identified, what improvements they can be done. So based upon that, in 1997, so OECD, the first major revision they have been done in 1997 as a principles, and there they have included the quality assurance and the study director roles, equipment, materials is what they have been using, test and reference items. So whatever the things is which have been missed in the early 1981 principles, they incorporated, and that is the first major revision which in 1997 they have made. Still, we are following the 1997 GLP principles with some revision changes and with some guidance updates and some amendments whatever they have made. So that still we are using it, and continuously from 1997 to till now 2023 also, there will be little bit amendments they have been placed, and some that they didn't do any major revisions, some amendments and some guidances they have been incorporated. So in later slides, I will be showing it in which areas they have been updated. So next slide. So coming to the principles, total 10 principles are there in the GLP. So the 10 principles will be seeing that major things is whatever it is there. So first is will be the organization and the personal responsibilities. So as per definition, already it has been mentioned that it's an organization process. So the major responsibilities is lies with the organization. So because they are the persons, they are going to give the resources, and they are going to give the facilities and what are the requirements is it is there. So the organization has to provide the necessary things is to continue the GLP practices in their organization. So they have to ensure all the things, and they are the person who is going to take the study director or QA. They have to do assign the QA, and they have to keep an archival facility, and they have to make necessary roles and responsibilities they have to do. So that is the major lies with the organizations only. And from that organization and individual persons, the study director responsibility is to have the individual responsibilities to conduct the study personal study how it has to be done. So the laterally every individual principles will be having the slide. And next one will be the QA. So that should be an independent QA unit should be there, and how the process it has to be done, and what is their activities they should be defined. And facility equipment and management. So here, how the facility it should be there, and what type of test they are going to do, and to that particular test, and what type of equipments they should have. So these all will be defined in that one. And next is the SOP, standard operating procedures. So SOPs and protocols to execute the study or to conduct the testings. So the SOPs, how it should be there, and what are the things it should be covered. So that it will be defined in that one. And next, we have to conduct the study. So how the study has to be conducted, and what are the data it has to be retained, how we have to maintain the data. So all are been defined in the study conduct and the data management. And once the study conducted, the final will be the reporting of the data. So how who the collected data results, how it is comes from the raw data, the report has to be generated. That report has to be reviewed by the QA, and once the report generated, it has to be get archived. Once after archiving, again we have to retrieve it periodically, and we have to ensure that the data has been still proper it is there or not. So there is no any degrading of the papers or the CDs or the tapes where they have been doing it archive. So whenever they want to retrieve the data, it has to be in easily readable way, and it has to be reproducible whatever the data it has to be done. So next slide, sir. The first principle is the organization and personal responsibilities. The main principles lies with the management. So they are the person, if for any companies, if they are going to open other laboratory, they are going to start, the management, it's the first to ensure that they have been recruited the proper leadership. So proper leadership, mainly the study director will be the main person for the contact persons. So study director should be there, and whether they have retrieved the proper, so they have recruited the proper study director who have proper experiences, and they have to appoint the QA unit, and the QA person also should have a proper experiences in the activity what they are going to do, and they have to ensure that the facility what they are going to arrange it is properly it is there or not. So these all is lies under the management. So once the management, if they recruited the study director, so the next the study director responsibility is to conduct the studies, or he is the single point contact for the any study, whatever they have been conducting it, and he is the responsibility to check, to recruit the study personals, and he is the responsibility to assign a principal investigator. So in multiple study sites, so if it's a different sites, if they do the same study, so it is his responsibility to contact with the individual sites, and to assign a principal investigator in each site, and his responsibility to ensure that so each and every multi-sites the activities are being done in similar way. So in one side they follow the different practices, and other side different practices, means the end result it may not be same. So the results it has to come same, means the procedure they follow should be same. So the communication between the different sites, everything it has to be taken care by the ensured by the study director, and study director's responsibility to recruit the study persons also, and he is responsibility to ensure that whether the activities are being done properly or not, and any deviations if it has been happen, so to the defined procedure or the protocol, so it has to be come to notice to the study director, and his responsibility is to see whether the deviations, anything if it has been happened from the protocol or the SOP, it's going to have any impact on the study data. So that if impact it has to be noted down. And he's without study director's signature, the final report cannot be submitted to the regulatory. So he will be ensuring that the data, whatever it is being captured in the report, it reflects the raw data. So that is the role of the study director. And study persons, so the persons who is going to do the actual activities or actual analysis or actual performing the activity, they are the study persons. So study persons also should be properly qualified persons should be recruited, and once they have been joined in the organizations, so for them the proper trainings has to be provided, practical training as well as the SOPs and all the trainings has to be provided, and their responsibilities is to ensure that they are being healthy they are there before executing the activities, because it's a non-clinical studies means it's all animal handling it will be there. So if the individual persons who do the activities, if he has been in sick, that particular his sickness should not get affected to the animals. So because of that, the results it may get vary. So the person who actually doing the activity, you should be healthy enough to do the activity, and health precautions is main. So they have to keep all PPEs to do this activity, and they have to follow the procedures, and they have to ensure that whether they before they are using any equipments or the instruments, all are within the calibration limits. It's there or not. It has to be ensured. So with the healthy precautions, if they do the study only, the integrity of the study can be maintained. So next slide, sir. The second principle comes a quality assurance unit. So QA, they are the independent unit, and they should be reporting directly to the management, and the person who is involving as a QA should not be involved in any of the study related activities, and their independent unit, and they are directly reporting to the management, and they ensures that the protocols or the activities or monitoring activities will be done by them. So the whatever the procedures or the details which we have mentioned in the protocol for execution of the study. So they will ensure that they follow as per the defined procedure and process, and they will be seeing with the regulatory requirements are being compiled with the SOPs and the protocols. So they are the persons who will be monitoring the study, and they will be reviewing the data whatever we have been given to them. So they will be doing it. Three type of audits will be performed. So one is a system audit. So system audit is so system wise the each and every areas is like how the test items have been stored, and storage areas how it will be maintained, and chemicals areas is how it is being maintained, in and out inventories is how the labelings it is being done, whether the calibrated instruments are been calibrated or not, installation qualification, performance qualifications are being done or not, if they are using computerized systems for generating the data, so those computerized systems, whether it is been validated or not. So, and if any systems is, equipment is, if it is comes under maintenance, so if they do maintenance activities, whether they did service reports and the calibrations are not perfect or not. So these are the documents, it will be verified by the QA. So that comes under the system audit. And one more is the in-process audit. So when the study is being going on, so they will come and they will be checking it whether the analysis are been performing, other the process are been done as per the defined SOPs or the defined method or the protocol how they have mentioned. They will be ensuring it is been done or not. So that is in process audit. And retrospective audit is once the study execution has been completed, that the laboratory will be providing to the QA the raw data. So the raw data they will be cross checking with the systems and everything. They will be checking it, and they ensure that the raw data, whatever it is there, that data is only it is reflecting in the report like that. So their statement finally it will be given for the report also. That statements defines. So the activities has been done as per the defined procedure and process, and as per the regulatory it has been followed or not. And the data, whatever it reflects is in the raw data, same thing only it is there in the report like that. They have to give a statement, and they have to maintain the schedule also. So each process, whenever they do the audit, so who has been done, who is the auditor, when it has been done, and what are the scopes they have been covered, and how much data they have been audited, how much percentage of the data they have been reviewed, and the durations, everything it should be maintained, and they should maintain the schedule also. So before start of any study related audits, so they have to assign the person, and that person they have to come and do the audit. So these all it is there related to that one, and again they have to maintain the master schedule also, and SOPs is also it's under their control only. So these are the functions of the QA, and coming to the documentations, they should have their own QA procedures should be there, and the inspection schedules related to the system, how frequently it has to be audited, or vendor qualifications if they give, if they are procuring the some items as like standards, chemicals or columns. So whatever it is there, it has been procured, the critical items which have been procuring from outside, so the vendors has to be get qualified. So vendor qualifications has to be done. They have to ensure that they from the defined protocols or the SOPs, whether any deviations happened or not. And they will be checking it, corrective actions, as if they will be having the trend analysis will be done by QA, and they will be checking, and any corrective actions to be done, kappa has to be raised or not. Those trackings will be done, and investigations, if anything is there, they will be checking it, and the trend analysis will be done. So these are the activities which QA will be doing, and they will be maintaining all the SOPs and the documents. And under QA only, the archiving facilities also will be there. But any archives, it comes under a different principles. When that slide comes, then I will explaining about the archives also. So next slide, please. So next principles is facilities, equipments, and management. So facilities, equipments, and management. The main responsibility is also this lies with the management itself, because the type of work they want to do for that particular type of work, they have to give the premises. So the premises also, it has to be checked. It should be properly bifurcated from the storage area, instrumentation area, process area, and wastage or area like that. It has to be properly bifurcated, and what type of work they do, based upon that, it should not be get interfered with any rodents or pests. That should be clearly away from that. And they also be ensure that the flood or earthquake, anything, suppose anything happens, as how they are going to save the things and from fire. Those things is also, it has to be taken into the consideration when they are designing the laboratory. So adequate space should be there, and the temperature, humidity maintenance, it should be done, and disposable activities separately we should have, and the storage of test and references items we should have separately, and there should not be any cross contamination should not happen. So that it has to be designed in such a way that that there should be a controlled environment, and separate zones should be there for each area how they do it. Coming to the equipment, so what type of work they are going to do, for that the equipment should be there, and how much work they are going to do. So suppose if they yearly do some 50 studies, if they do, they should have equipments and the resources to execute that 50 studies. And they get 50 studies, and they have only a resources for conducting 10 studies, then that it is not been accepted. So the equipments also in such a way it should be there, and what type of work they do, for that type of equipments they should have. If like LCMS, if they want to do it, LCMS analysis, they should have LCMS. If they should do elemental testing something it is there, then ICP-MS should be there. And type of work what they do, as for that requirement, the equipment should be there. And before that equipment's installations, so it should be ensured that IQ, OQ, PQ should be done before the equipments are been released for the usage. And each and every equipment they should have an SOPs. SOPs should be properly in place, and the before handling of the equipments, training has to be provided to that persons, and SOPs should define the frequency of calibrations, how frequently it has to be done, and what are the calibration parameters it should be done, so that it should be mentioned in the SOP. And if the equipments have been handled with the help of a software, that software before releasing computer system validation has to be done, and URS has to be there before when you are getting the equipment. And based upon the usage of that particular instrument, for that software, what we expect it and what we do it, as per that the CSV protocol to be prepared, and the software validation has to be done. Once after everything, if it is been done, only the equipment will be released for the usage. And once it will be released for the usage, preventive maintenance frequency it should be defined, and maintenance if they do anything, so that it has to be captured, and the service reports, everything it has to be captured. And coming to the test systems, so the test systems is the system, the test items which we are analyzing comes under the test systems. It can be, suppose if it is an animals studies if you are conducting, then animals is called as the test system. So suppose if we are doing pesticides are related to the agriculture, it is the plants, everything it will be considered. So cells, bacteria, analytical instruments, and animals. So these are called as the test systems. Where the test items we are going to analyze is called as the test system. So this test system should be properly bifurcated from the test items. So test items is the items which we are going to under investigations. Those are called as the test items. So any drug or any chemicals or any solvents or any items which we are going to investigate in the test systems are called the test items. The test item and the test systems, both it should be properly separated and no cross contamination should be there. So in such a way, the facility and the equipment should be placed and properly it should be managed and design has to be done in such a way. Next slide, sir. Next comes the standard operating procedure, as well as protocols also it comes under these particular principles only. And standard operating procedure is it will ensure the consistency, reproducibility, and compliance across all laboratory operations. So multiple study persons will be there, and all will be doing their individually. They will be doing their activities. So each and every persons, if they involved in the same studies, then all have to do in similar way. It has to be done. So the way how it has to be done, the procedure, process, everything are been defined in the SOPs. So the SOPs it defines the procedure and the roles of the persons who are going to involve and what activities they have to do. It should be there. And if the SOP has to be done, there should be a proper change control procedure should be there. And how the change control procedures it has been done, and it should have a historical revision history should be there, and first revision to the second revision, what are the changes has been done. So those also it has to be captured, and the chronologically order the SOPs procedures has to be defined. So first, second, third, fourth step. So how the stepwise it should be there in such a way it should be there, and no ambiguous. So if they read that SOP, so for all the persons it should give the same meaning. So if non-ambiguous way if it is there, so they read it and two different meanings it should not be given. So in such a way the protocols has to be defined and it should be in such a way that all has to be understand clearly, and after reading that SOP, the procedure and process execution should be inconsistent, and if they do it repeatedly, the reproducibility has to come. So for that purposes only SOPs has been done. So SOPs will be developed by the subject matter experts. The draft first they will be doing it, and that draft it will be given to the QA for the review. And the QA once they review it through these all will takes places through the change control process. And once the QA review it is been completed, then it will go to the management. So all the SOPs has to be approved by the management. And next it will be the implementations. Once SOPs has been identified and it has been prepared, then before or it's going for the implementations, the training has to be provided to the all the staffs. So for training, there should be a training attendance should be there, and individual training record should be there, and evaluations of the particular SOPs. So questionnaires also will be provided, and evaluations also will be done. And after that, persons has been qualified by getting a mark in that particular SOPs, then the particular SOP will be get implemented, and the after getting training only, that particular staffs can be involving in the activity of the SOPs. So whatever the SOPs is there, so all the persons it has to be reviewed. And in SOP, once if it has been reviewed and it has been released for the usage, so we should define how periodically the revisions has to be taken. And it's only for periodic revision or during any other changes it has been done in middle for the improvement of the system or any changes in the guideline, then they have to take the revision of these SOPs. So this all it has to be done through the change control, and these documentations, everything, it will be controlled by the QA. Next slide. So the protocols and in SOPs, already I told protocols also it will be involved in if you are developing the protocol. So the protocol should have the title, proper title. It should be there, and the protocol has to be have objective of the design. What is the design it is there, and what is the objective of the protocol, and who is the sponsor, and where is the test sites, which sites we are going to conduct the test, and what is the design we are going to use it, and what is the species or the age and race, what we are going to use it, and what is the dose we are going to use for the based upon the body weight, how much we are going to use it, and what is the designing of the study, and how it will be executed, and what type of analysis will be done, and how the analysis will be made, and finally how the results will be interpreted. All these details is everything it has to be captured in the protocol. So once all these protocols, if it is been done, then it has to go for an ethical approval. So if it is a animal studies, animal ethical committee, they have to approve that one. So if it is in a human means, ethics committee approval has to be done. So whatever the protocol it is there, so it has to be get approved from the ethics committee, and then only the protocols after that, once it has been get approved, it has to be training has to be provided to the persons who will be involving in conducting of this study. So once after training it has been completed, then the protocol, as per the protocol, the executions will be happen. When the execution is been happening it, so it has to be recorded. So the data, whatever we do it has to be recorded. So the recording has to be done as a raw data. So here it comes the ALCO plus principles, so it has to be taken care. So like entrepreneurs, attributable, legible, original, accurate, and other few things is also it is there. So all it has to be followed when we are being reporting or documenting or recording the results which we execute during the study. And once this raw data has been recorded, so then next it comes the data integrity. So the data, whatever we've been generated, either it's raw data or it's an electronic data. So how secure we are going to store it, and in instruments if we are using it, so how the audit trials are been enabled, and based upon the designations and roles, how the access controls has been provided. So the delete options should not be given to the users, and because delete or overwriting option should not be there. So if it is there, so they will be deleting the something, suppose if there is some obvious results, if it is comes, or the results which they didn't expect, if it is comes, the analyst they have an rights to delete, means they will be deleting that one, and once past one only it will be there. So like that it should not be there. So whatever the data it has been there, so everything it should be captured in the instruments, and who has been done and what activities has been done. So it will be captured only through the audit trails. So each and individuals they will be provided with an username and the passwords, and passwords should not be get shared. So the activities, whatever they do through electronic signature, if they do it, we will be able to identify who has done that activity and when it has been done, that it should be able to clearly identified. And once the studies, everything it has been done, the backup, it has to be done. So either the raw data keeping in archives, or the data which has been generated in the instruments, it should not be get lost. So daily backup data, or weekly, yearly, monthly. So different processes, it should be defined in the SOP, and the backups also it has to be done properly. Why the backups are being done, that we'll be doing analysis. If anyone lost due to any circumstances, or any short circuit or anything malfunctions or any failures of the equipment, then the analysis, if there is no proper backup, the data will be get lost, and we are can't able to justify. So what happened, the results is like that. So each and every single records of the data sets, it has to be properly backed up, and the prevention, how we are going to do all these things is backup details is everything, it has to be maintained. So the loss has to be reduced. So backup, and once backup if it is been done, so we have to ensure that the backup the data also, whether we can able to retrieve it and restore the data, and we can able to see the data which have been we have archived it is long before. So that insurance also they have to do. So for that also they should have an SOP. Next slide, sir. So once the report we have been done, the recording has been done and QA monitoring it has been done, then final report has to be prepared. So as I earlier I told, study director signatures it should be there, and he will be ensuring that the results are whatever it is there in the data it matches with the results or report or not, and deviations anything it is there. So he has to ensure that deviations has been recorded, and the assessment impact analysis has also been reported or not, it has to be checked. And without study director sign, the report will not be get finalized. And once the report has been generated, so it has to go for archiving. The archiving also protocol. So archiving, if you see, what are the data has to be archived is that, so related to the equipments and the instruments, if you see, the CSV documents and IQ, OQ, PQ document, calibration documents, and environmental monitoring, the temperature, humidity, whatever we do, that it should be archived. And the equipments will be having, each equipments will be having usage logbooks will be there. So those logbooks has to be archived. So any equipments is there, so it will be having a maintenance logbook. So that maintenance logbooks, it has to be archived. And computer maintenances, whatever they do it, that details also has to be archived, service reports, and these are the things that it should be archived along with the study data also. So archiving also, they should have an archivist, as they should from QA only usually archivist will be assigned. So the archivist will be a separate role, and many persons should not be provided as an archivist. They have to appoint a single or two persons as an archivist should be there. And before going to archival also, so the reason for entering inside the archival room and access why they have been provided to entering in the archival room. So those entry exit details also has to be captured by the archivist. And the retention in the SOPs also we have defined how many years the each documents has to be stored. For individual persons, we see the JD, job descriptions, CV, individual training records. These also has to be kept in the archives. So these all archives has to be retained for related to the equipment and studies, whatever it is there. So 15 years at least it should be there. Other than that, individual person's training records until lifetime, the archival has to be maintained for that one. So suppose if archiving, if it is not facility is not been sufficient, and if they go for third party archiving, so they also should ensure that how they made an agreement and how the samples files have been moved and how frequently they can able to take the files, and if they give request and how many days they can able to get it, and what is their scope and what is company scopes, all it has to be defined. So that it has to be maintained properly for the documents whatever we have generated. And next comes the access control. So control retrieval procedure, once if it has been archived, so if during audit time or during any inspections, if it is comes, so at that time if they ask for the retrieval of the data, those control retrieval data says how we are going to retrieve it and who has to be and what purposes the documents can be retrieved and what is the procedure and process to retrieve the data. And once if you retrieve the data and if you do any changes to the existing files or data, so how we are going to address that issues is, and if the data is archived, then re-archival procedures. So how many days the data has to be returned to the archives, and no changes if it has been done, how we are going to declare it. So these are all the things it has to be captured in the SOPs, and access should be controlled. So that it has to be detailed in the SOPs, and the process also should be there. Next slide. So that comes the retentions of this one, as well as if it is in retention, one more principles retention of the data as well as the samples also will be there. So wherever the test items is, we have been storing that investigational products. So how far we are going to store that particular products. So that also it has to be mentioned. And for that temperature, humidity maintenances and temperature records also has to be archived. And now whatever we are seeing, all the principles we have been gone through for as per the GLP guidelines. And this GLP, where we are applying it is in for pharmaceutical products, is whatever we do for the INDA, NDA, ANDA submissions, whatever we do. So non-clinical safety studies is we have to follow this GLP. And for agrochemicals like toxicological environmental studies, pesticides, agricultural products, so those things also we have to do the GLP laboratory environment only, it has to be done. And industrial chemicals, so where this registration, evaluation, authorization, restriction of chemicals from European Agency, Chemical Agency, and Toxic Substance Control Act as per US. So they also, for any industrial chemicals, it should be get marketed over there. So they have to get approval from these agencies, and that test, whatever they do, it has to be done under the GLP compliant laboratory only, it has to be done. And it comes to the medical devices also. So the medical devices also comes as under the GLP. So the testing of medical devices, wherever they do the testing, so that particular lab also should be a GLP lab. So that GLP lab, if they do that medical device test, it will be get approved by the regulatory. Next slide. So the emerging areas and GLP adaptations, so where the GLP is still they are planning to implement is that, so nanomaterials is where the nanoparticles is we will do it for that characterization, and like size distributions, surface areas checking it, and characterizations of nanoparticles is how to do the characterizations, how it is being done correct or not. So what are the SOPs they have for reference materials from where they got it and how it has been done. So they are planning to implement the GLP in nanomaterials areas characterizations also, as well as the biologicals and ATMPs. So ATMPs is advanced therapy medicinal products. So where the gene therapy, cell therapies, and the tissue engineered products is to do it. So they are doing it. And biologicals is that the medicinal products is which we have been developed from the living organisms are the biological sources. So where they do the biologicals, and ATMs, they are planning to implement this GLP also. So when they do the testings, those analytical techniques and validation parameters is the GLP, it should be there like that. So they're planning to do implementations in this particular area, and also in silico methods. So in silico computational toxicological models, so where they don't use animals, without using animals, they'll be checking it, and in silico models which will be the best suit and for which receptors it will be acted correctly. So that areas is where they will be using the softwares. Those softwares is whether it has been validated and it is the results which they give, whether it will be correct or not, right? And that algorithms is what they have been calculated, the formulas is whether it is correct or not. It has to be ensured, and the training data and the how they do the activities is these all also OECD guidances. They are planning to implement the GLP in this particular area also. Next slide. So that is the applications and where they are emerging it. And one, two, three practical examples is so how the GLP studies will be executed is that. So once we see it for the toxicity study. So first, before executing the toxicity study, the study design has to be done in the protocol. So the protocol, it has to be defined. So what is the purpose and scope of the study we are going to do it, and how the dosings will be given, how blinding the studies, how the animals will be, the dosing person will be blinded, double blind, single blind, or open label. So that has to be defined. The dosing, how much weight dose they will be giving based upon the weight of the mice or rat or whatever the species they selected, and what activity they are going to check, and finally endpoint on how the analysis will be, observations will be made. and it all has to be defined in the study design. And once the study design, it is protocol has been done, and the sponsor has also given approval for this, then ethics committee approval has to be done. So once there is protocol approval is over, the executing study persons has to be get trained for this particular protocol. And once the training has been over, so they have to perform as per the planned design. So there, the dosing records, it has to be maintained. So how the species has been selected, and what are the species and what weight it has been there, and the dosing, how much they have been done on the weight, it has to be captured, and clinical observations. So how the differences it is being shown, there is no differences, all it should be documented. Once the study execution has been completed, necroscopic findings is so once the animal done, so there will be sacrificed and they will be checked for any cancer related activities has been happened, and why the animals has been dead. Suppose if any dead animals happen, the reason behind the death it will be has to be identified, and histopathological sites and the chain of custody from handling of the animals from segregating it, dosing, and doing observations how it is and to final end. So every chain of custodies, the documentation has to be maintained. And in these phases, the QA in middle, they will be doing the monitoring activity. That is protocol stage itself, they will be involving. So protocol will be audited by the QA, and when they do the activities like dosing and checking the observations, life phase inspections, in life phase, that is inline process inspections will be done. Once after completion of the entire study, the data verifications before finalizing of the report will be done. Once the data verification is completed, then the report preparations will be started, and the reports will be reviewed, and they will be cross-checked against the raw data, and finally it will be verified, and the data will be submitted to the regulatory. And next slide. So this is a practical example for the environmental pesticides registrations if they want to go. So how they will be doing it as it's related to the environmental. So the studies will be conducted in the open fields. So in different areas they will be conducting it. So different field sites they will be going it. And from there the samples will be collected and it will be sent to the analytical lab. And analytical GLP lab, they will be doing the test, and then finally in different places analytical labs if it is there. So different places if they do also, the method should be remain the same, and overall that the data sets it has to be kept it in a single data management particular places it has to be placed. And data integration says how it has to be done. See, during the analysis, the different stages they will be checking it. So hydrolysis of samples, and photolysis, soil absorption, metabolite identification results. These all different places, whatever they do that, different studies, all it will be combined together, and they will be keeping as a single study master file. So different places, different tests if they do it also. So all together it has to be integrated, and it has to be placed in a single place, and that it has to be placed in a single, and that we have to submit it for the regulatory submissions. So if it is related to the environmental, so for US it is an environmental protection agency is there to approve the environmental related studies or the materials or whatever the chemicals they use it, that they will be giving. EFSA is a European Food Safety Authority from Europe. So they will be evaluating the study submitted data, and they will be checking all the data, and they will audit it, and they will be giving the approval for the pesticides registrations. Next slide. So this is for preclinical biologics if they do it. So the complex systems is that the GLP application will be done for the immunogenicity assessments and bio distributions, how it has been happening it, and toxicity is example is a monoclonal antibodies if they do take it. So where the GLP implies is assessing the immunogenicity and biodistributions, how it is there, and toxic kinetics, absorption distributions, metabolisms, and how it will be happen. This all the analysis things is the GLP involved. So for this testing, the method validation, what they use it in bioanalytical, so it has to be the validation has to be done as per the ICH guideline, and all the parameters which is being done are correct or not, it has to be ensured. And as per the guidelines, the validation parameters if it is been done, and QA they will be checking it and auditing it. Once the validation, if it is been completed only, the samples will be given for analysis. So here the main thing is that the study director he has to get a coordinations between the pharmacologist and the toxicologist and the lab where the analysis will be happen. So everywhere the single point contact will be the study director. So he will be ensuring it the work has been done properly and the documentations has been received properly, and communications and protocols and training between the different sites has been given properly or not, it has to be ensured by him. Next slide. So this comes the guideline revisions and amendments and how it has been changed from past to now. That be based upon the scientific advancements and the changes which has been happening in the because it's the guidances has been released in 1997. So from 1997 to till now, there are a lot of changes has been happened, and as a scientific as well as in technology wise also there is an advancements have been done. So different advanced equipments came, and identifications of metabolites or identifications of impurities also has been increased. So based upon that, new methodologies and how to update the validation criteria has been done. And coming to the digitalization, technology evaluations, and involvement of AI tools and automation necessity in some cases, and computerized system, how we are using it to generate the data. So these also the changes is also it has been came, and regulatory convergence. So they when they do auditing it, and different regulators they will be having their different approaches will be there. So these all put together, and they have some discussions as it has been done, and that also they have put it as on some because of that also there was some revisions and some amendments they have been done, and inspection insights when they go for an auditing. So they identified some deficiencies and they get some clarifications as why it has been happened. And when they go for an audit, the responsibilities of QA and the data integrity, these all are the factors which they have made amendments and the revisions amendments to the guidelines. So 1997, as I told, it has been guideline has been done, but major revisions they have not done for the principles. Only amendments based upon the current technology or current regulatory how far they have been identified, and from that one and based upon the equipments, now the changes have been done. So these are the drives which they have made changes to the guidelines. The next slide. So the updates, if you see, these are the updates as they have been done. Actually, in 1997, once after it came, the second revision they have made it in 2002. 2002, what they have done is that computer systems they use a lot of computer systems rather than the manual documentations. So because of that, in 2002, computer system and electronic data handling they have been introduced. In 2004, they identified the multi-sites they do the studies and how QA has to involve in the inspections in that multi sites, and each sites if they do it, how the data has been traceable, how the data management it has to be done. So that they have been defined in 2004 and 2016, like 1970s, the GLP how it came in 2016. Before that, in 2010 afterwards, a lot of companies they do some falsification of data, testing of injections, trial injections they give, audit trials they are switching off, access controls are not been properly defined. So because of that, data integrity has came, and this data integrity each and every regulator says they have released the guidances. And they brought the importances of audit trail, electronic signatures, QA risk management. So based upon this, the 2016 again they have made some amendment and guidances for data integrity, audit trail, and electronic signature. And 2021 they have made the risk-based QA system. So how the risks analysis is, so how the change control it has been done, and how the user access, how it has been provided, and who is the administrator for any software related things, and audit trails should not be disabled. How active the audit trails is there when they're conducting the study. So that and all it has been came in 2021. And 2020 independence of the study independences, like multiple sites studies it is there. So each study there should be a principal investigator it is there, and what is the role of this principal investigator and how he is going to see the different multiple sites. So study independences they have made it, and the involvement of the sponsors they have been reduced. In 2020 they brought that particular amendment, and 2019 also the guidance for reviewing of the GLP status. So they brought reviewing of GLP, how it has to be reviewed, the status that they brought it. In 2018, if you see, test control, test material control. So how we have been arranging the test items and how to use the test items. So those they brought it in 2018. And 2022, the revised the guidance for the validation acceptances, and based upon the new equipments, how the test methods has to be done, and how internationally we are going to accept it. In 2023, what they brought is cloud data management. So cyber security and cloud data management they have brought it. And these are the recent updates which they have been incorporated in the OECD GLP from 1997 to 2024. Next slide, please. So recent updates in the FDA and EMA regulatory changes is that so 2023 US FDA draft guidances they have given for electronic source data in GLP studies. So they brought Alcoa Plus principles 21 CFR Part 11 for computerized systems. So username, password, electronic signature, reasons for any changes. If you do any alterations, if they do any modifications, it should not be get overwritten. So the original data and the new data, both it has to be there. So like this, so many changes is related to the electronic data generation and maintenance of the electronic data generations. They have brought it in 2023 by US FDA, and 2022 EMA. What they have made it for the advanced therapy medicinal products is how to do the trials, clinical trials, as when they use this advanced therapeutics, like already I told, like gene therapy or cell therapy, and medicinal devices which are linked with this gene therapy or cell therapy, how it has to be executed the GLP compliance with GLP compliance. So they have updates on this particular region, it is ATMPs, and ICH S12. So ICH S12 is a non-clinical biodistributions of therapy products. So there, how the GLP procedures, and it has to be technical requirements and what specialization, how it has to be handled. So like that, the regulatory they brought updates is, and these areas is also they want the GLP compliance to be followed. Next slide. So digital transformations and the GLP compliance. Now the digital transformations from manual logbook entries or manual method notebook entries, it has been improved to the electronic laboratory notebooks. So there because the falsification data are backdating, everything it will be get erased when they have started using the laboratory notebooks. So where the activities, whenever they do it, like LIMS systems, so everywhere it has been implemented. So real-time monitoring of the data source will be done, and backdating it or back signing it, or the availability of the persons should not be will not get manipulated by using all these techniques. So these new techniques is also they have been using it for doing the validations, and audit trails is also it can be easily we can able to trace it, and who did what time, what activity, and reason for any changes is it can able to identify it. And alcohol plus principles. So that is the major thing for the data integrity thing. If these principles, if we have been following it, so the data integrity will not happen. And clouding of systems. So cyber security attacks are back. So whatever we do that, any studies is the data is important. So that particular data, it should not be get destroyed, or it should not be get attacked by spams, anything. And so how we are going to save the data, whatever we generate, and suppose anything disaster happens, and what backup recovery plan we have, and how the business continuity plans it has been there, backups how we have been storing and restoring it, and this all will comes in the cloud systems. So to avoid all data loss, now they have been recommending to store all the data sets in the cloud system. So each and every single recording also it will be go to the cloud systems. So it will be easy, irrespective of any geographic or irrespective of any location, whether flood will come, or earthquake will come, or some other things if it has been happened. So the data loss will not be happen as all the data sets it moves to the cloud system. So the recovery of that data is also very easy. It will be there, and backup redundancy will not be there. The next slide. So the common inspection deficiencies and remediations is when they do the GLP inspections is that the top inspection findings is that incomplete SOPs, is the inadequate training of the study personnel, and missing of equipment calibration logs, and QA was not, they are not independent, and they have been getting influenced by the technical persons or the management, and raw data are non-contemporaneously recorded. So online documentation is not there. So they do activities and they do the document in letter, and sometimes they do complete the activities and go and sit and do the documenting. So that those things are the top inspection findings from the GLP inspectors. And if they do for any failures, if it has been happened, and when they go for an investigation, and they're not doing it proper root cause analysis. So the root cause analysis, it has to be done like fishbone, that 5 whys it has to be done. So why, when, where, people, whether it is due to people or process or technology or the environment. So based upon this, the root cause analysis from each individual thing wise, we have to segregate and we have to do. So this is the remedial action for the findings. So root cause analysis doing by using 5Y and fishbone technique, and kappa strategy. So we have to check any repeated failures, if it has been happening it, the trend will be taken it, and during the management reviews, and it has to be decided. So proper preventive action, how it has to be done. So any preventive actions, if we have been documented, we have to go for effectiveness check. So effectiveness, how many days we are going to check whether the preventive action what we have defined, it has been effective or not. So that effectiveness check has to be done. And the main reason behind for implementation of kappa is that to stop the reoccurrences of the failures or the events which will be happening it. So that it has to be properly checked, and then it has to be involved. So by doing this root cause analysis and kappa strategies, is the most of the deficiencies which are being faced during the GLP can be get reduced, or it can be fully, it can be removed. The deficiencies can be come over across that. And next slide. The best practice for GLP implementation is it's leadership commitment. So it comes from the management. So everywhere the cultural quality culture has to be promoted. So as per GLP, so every year in GLP companies they have to provide the GLP training to be provided, and that way the management will ensure that they have been imbibing the culture in each and every individual persons, staffs to involved in that one, along with the data integrity things is also they will be doing all corporate principles. So these things will be reducing the integrities related issues, and it will increase the performance of the lab. So it's all based upon the commitment of the persons, and it will improve the quality culture and management, how they are allocating the resources. Based upon the resources allocations, the performance also will be increased, and risk-based approach. So prioritizing the validation, and we will be checking it. Gaps, it has to be identified, and each gaps, whether we have to identify where the gaps are there, and whether it is a system related thing or a human related thing, those has to be identified, and based upon the criticality, the risk has to be sorted out, high, medium, low. So like that, it has to be checked it, and based upon the high risk, the implementation of the kappa, it has to be done, and there we can reduce the problems, and we can able to monitor how that kappa has been effectively doing. So this way we can reduce the impact of the data what we've been generating. And continuous training. So continuous training to these staffs, it has to be done. So because there is an changes, it has been taken from, see, if you see the GLP guideline, 1997 the guideline came, but 2024 up to that, there are a lot of changes is there based upon the upgradations of the equipments, automations, and computerized environment. Based upon that, all the industries, they have been working to improve their lifestyles as well as their mode of process, everything. So to that one, they have been changing all the guidelines, and they are providing supplements, everything for these principles. So to that, if they should come to know by continuous training only, there will be an improvement, it will be there. So refreshing training should be there, regulatory updates will be there, and based upon the regulatory recent audits, they may implement few things. So those, all the things, it has to be explored to the staffs, and continuous training if you give, and the assessment of their competencies will align with the regulatory updates, and it will improve the practices of GLP. Next slide. Future trends and the strategic considerations. So future trends, how it will be there, AI and machine learning. So wherever the toxicity studies or endpoint studies we have been doing, and predictive modes like QSAR or in silico models is what we do it. So there the validation framework, how we have been going to do it, and GLP's implementations, it will come in that particular places, and automations and robotics. So many places automatic processes has been came. So in that automated process, so how the protocols have been followed, how the documentations is been done, and how are the performance of the robotics automations instruments, how far it is going to give the best results. So there it has to be GLP will be coming, and that will be implemented it, and GLP compliant recording of the green chemistry matrices and waste reduction practices. So sustainable documentations like now recent green chemistry, all everywhere it has been increasing it. So there in line with the green chemistry, how the GLP compliant it can be done. So there also the implementations of GLP will be there, and obviously it's a global harmonization. So each and every countries who are been the members of this one, or even though they are not member mutual acceptance data if they have been signed. So if you do the activity in single lab, that it can be utilized in throughout the world. So that is the main concept of the GLP. In GLP lab, if you do it in one country, that particular product, it can be go for the sales, or it can be sent to the other countries also, and other country regulatories also, they will be accepting the data which has been generated in any of the GLP lab. So there the money will be get reduced, and the studies conducting will be get reduced, and the product which will be released to the particular regulatory will be get increased. So it's going to increase the economy of the particular countries. So that is the main motto behind the GLP. If GLP approved laboratories, all the activities has been done. So that is going to reduce the more burden to the companies, and the products it will be getting easily available to the humans, and for their health it will be useful. Next slide, please. So from this presentation, the key takeaway is that the principles of the GLP. So the principles of GLP, it has been when years this goes on. So the amendments they have been making it based upon the how far the world it is getting improved, like in computerized way, or other things the technology wise. So the principles, as per that, each and every areas of principles they have been incorporating the advancements, and they see that the quality of the data which has been generated, it has been correct, and there is no impact on the study when they conducted it, that will be checked. So main principles like study director, QA, and document management is Alka plus principles, and Kappa management, and cyber securities, and backups. So these are the retentions of the data. So these are the major principles. All the principles they remain the same, but when the days goes on, the technology is getting improved. The guidances and the supplements also, they have been including it, and how far it can be controlled and how far it can be monitored. So they do the implementations also. So these principles, if it is being done, then the particular drugs or whatever they do the development, that it can be easily, readily, it will be available to the other countries is also. The time taking to reach product to the human, it will be get reduced, and the amount what they are going to spend on the studies is on the animals or the humans also will be get reduced. So these all principles, if it is being followed, or the regulator is going to accept the data very easily. So this is the principles which you come to know, and these principles will be helpful each and everyone when they go for the laboratories and they do the work in the labs. So this is highly important. So labs which they go for regulatory submissions, all they has to follow this one. So this is the basic things which they have to be followed, and they should know when they enter into the companies, and that will shape them to be the better person when they do the work in the laboratories. Next slide, please. Thank you for giving this opportunity. Please, any questions if it is there, please, I am ready to answer to your questions.
Participants, any query?
Thank you very much, sir, for that highly informative and insightful session. Your valuable expertise has certainly deepened our understanding of good laboratory practices and their significance in research. Now we'll move on to the interactive query session. This is a great opportunity for participants to ask questions, clarify doubts, and interact directly with our expert speaker. I encourage everyone to actively participate and make the most of this opportunity. Anybody, please? Sir, good morning, sir.
Yeah, good morning.
This is Mahira, Associate Professor from Department of Pharmaceutical Analysis. Thank you for your in detail presentation, sir.
Okay.
I have a doubt from which I used to face from my student side repeatedly. Is
Okay.
there any specific certification course on GLP offered for students outside if they are not working in any industries so that they can procure the certificate and go with that for any job opportunities?
Madam, GLP certification course it is there, but I will share the details. There it is there. GLP certifications courses is there, and if you go for US FDA site also, so US FDA also they have a means course like it is there. So half an hour to 1 hour we can go through, and they will be having set of questions, and then the sort questions can be answered. And once if you answered, finally they will be giving you the certificate, that the GLP certificate like that they will be giving. So in US FDA website also it is there. But apart from the US FDA, other GLP courses also they are providing it online. They do it and they provide the certificates. But for easy means you can go with the US FDA site, and then the course can be done. It can be done 1 to 2 hours. It can be clear that one. And apart from that, individually, some academics is also they are conducting GLP certifications courses online. They do it, but for that they will be get charged. But if you want that particular thing, is means let me check it out and I will share the details.
Yeah, sure, sir. Thank you so much. Thank you, sir. Thank you, ma'am. As we approach the conclusion of today's program, it's time to express our sincere gratitude to everyone who contributed to the success of this event. To deliver the vote of thanks, and our invite Ms. Aarti ma'am, Assistant Professor, Department of Pharmaceutics. Ma'am, welcome you, ma'am.
Good afternoon, everyone. It is my privilege to propose the vote of thanks for today's skill development program on good laboratory practices, the backbone of reliable research. On behalf of the Faculty of Pharmacy, Dr. MGR Educational and Research Institute, I sincerely thank our respected management, founder, Chancellor Dr. A.C. Shanmugam sir, President ER ACS Arun Kumar sir, Secretary Thiru A. Ravi sir, and Vice Chancellor Dr. S. Geeta Lakshmi ma'am for their constant support and encouragement. I express my heartfelt thanks to our beloved principal, Dr. N. Harikrishnan sir, for his continuous guidance and motivation. On behalf of everyone present here, I extend our sincere gratitude to our esteemed speaker, Dr. R. Karthik sir, Head Bioanalytical and Analytical Department, Citus Pharma Service Private Limited, Chennai, for delivering such an informative and valuable session. Your insightful explanation on a good laboratory practice and its importance in ensuring accuracy, reliability, and quality in research has greatly enhanced our understanding. The way you connected laboratory practices with real-time industry requirements has been extremely beneficial for all of us, especially as future professionals in the pharmaceutical and research fields. I would also like to extend my sincere thanks to our respected head of the department for the constant support and encouragement in organizing such useful academic programs. My appreciation also goes to the organizing committee for their dedicated efforts in making this program a success. Finally, I thank all the participants for their active participation and interaction throughout the session. Once again, we express our sincere gratitude to our respected speaker for sparing valuable time and sharing your knowledge with us. Thank you all.
Thank you. Thank you.
Thank you, ma'am, for the
Thank you, Mr.
heartfelt
Karthik,
vote
sir.
of thanks.
Thank you, sir.
Today's program has truly been an enriching learning experience for all of us. The insights shared will undoubtedly help us strengthen our research practices and academic pursuits. With this, we formally conclude today's skill development program on good laboratory practices. Thank you all for your valuable presence and active participation. Have a wonderful ahead. Thank you all. Thank you, sir.
Thank you, ma'am.
Thank you, sir.
Dear participants, please fill the feedback form.